Hi, this is Dr. Dan Guerra, and coming to you from Authentic Biochemistry Studios, still in the Pacific Northwest, but now in the state of Washington. Um, what I'm going to do today is take a trajectory to give you a foundational architectonic, word I like to use a lot. So, architectonic is basically a global pattern of existence that reveals kind of like a table of contents, precisely what you're going to find in a volume. And the volume or the corpus we're looking at is basic biochemical literature in the large sense, but in the much more narrow accommodation that we're dealing with here with autoimmune diseases, with ceramide as a mediator of the inflammatory response, something I've been talking about a great deal in authentic biochemistry audio lectures, and a few of them of late also in video. And really, it's all ever since January, I've been dealing with autoimmune diseases. This is quite a long arc, and we are not finished. So I talked to a couple of people who have seen these, and they said, gee, it would be great, Dr. Guerra, if you went back and talked a little bit more about endocrinology, because sometimes you talk about hormonal regulation and receptors, and it would be nice to know what is the general physiology and physiological biochemistry behind some of these aspects. And that's what I'm going to do today, as well as, of course, discuss current papers. So this is the title, what I'm going to give this is Endocrine Regulation of Metabolism. I'm Dr. Daniel Guerra. This is Authentic Biochemistry. Today is indeed the 17th of April, not 2019, sorry, 2020. So it is, the date is wrong. I'm sorry about that, but it's 2027. It's a Friday. All right. So there I am on that part of the slide. And there's my Facebook page. And there is the Authentic Biochemistry feed. On Facebook, you can see all these video lectures, which are coming through a YouTube feed. And then, of course, the anchor site is the general home for the Authentic Biochemistry audio. There's my email. If you want to contact me directly, I'll be glad to talk about any of the content in any of my lectures, as well as to consult with you in anything you want to discuss in biomedicine as it relates to published scientific research. And so cover a rather broad range. I've been teaching biochemistry, general biochemistry, lipid biochemistry, cell biology, cell physiology, molecular genetics, pharmacology, pharmaceutical sciences, mode of action of drugs. I also taught a course in nutrition not that far back, molecular pathology, pathophysiology over the years in medical school settings and in nursing school settings, as well as, of course, in graduate schools. So that's my background. I've done quite a bit of work in the area of teaching, but I've also done a lot of research, and I've also published in neuroscience and done some work in that area. So that's my background if you haven't watched me before. So let's get going. All right. So these are basic fundamentals of physiology. So let's take a look at what happens to glucose, amino acids, and lipids or fat by various tissues in a well-fed state. So well-fed state means postprandial, right after a meal, or indeed with people that have a lot of meals, people that are well-fed in general, okay? And this is a generalization. And I want you to get the notion of what's going on with various organs. So the beta cells of the pancreas are going to be releasing insulin because there's going to be glucose in the diet. The gut then is going to have glucose coming in and protein coming in and lipid coming in. The glucose and the protein, first of all, glucose is usually coming as a polymer like glycogen or starch. It's broken down to individual glucose units. And the amino acids are coming from dietary protein. The lipids are coming in usually as triacylglycerol, sometimes as phospholipids, sometimes more rarely as free fatty acids. And all of that in the lipid component is going to be entering the lymphatics, and it's going to always be associated with a lipoprotein. The initial phase of this process is association of triacylglycerol mono and diacylglycerol, some free fatty acid, some cholesterol, and some phospholipid bound up in a lipoprotein called the chylomicron. Okay, these are non-covalently bound lipids to protein. That's what lipoproteins are. So we have trafficking a non-water soluble component like a lipid. Of course, the sugars and the amino acids, the glucose in particular, and the amino acids are going to be making it directly through the portal vein because they are water soluble. So where they all end up is in the liver. Okay. Now with the lipids, there's a circuitous route, which we'll get into, much more detailed than what happens with glucose and amino acids. But let's take a look at what's going on up here. Here's your liver, major organ in the body. It is basically involved in distributing everything, the dietary that's coming from the intestine. And then also it's storing some of that. For glucose, it's storing it as glycogen. For amino acids, storing some of it as some protein. But primarily, it stores also lipids. So it can convert glucose and convert amino acids into triacylglycerol. And also, of course, phospholipids. And so your liver becomes a major component to redress all the dietary intake, lipid, carbohydrate, and protein, and then transport it when some of that is converted to lipid via the very low density lipoprotein, the intermediary density lipoprotein, and the low density lipoproteins. All of those lipoproteins are going to traffic outside of the liver. It's a much larger discussion. You can see here how glucose is converted in glycolysis to pyruvate and then to triacylglycerol. Triacylglycerol is then sent around to various organs such as muscle tissue and adipose tissue where it's stored. We store depot fat or depot triacylglycerol. You can also see that there is some of the glucose is used to store glycogen, as I said. The amino acids are broken down sometimes all the way via transamination reactions to the TCA cycle. Prior to that, some amino acids can be converted directly to pyruvate. For example, alanine can be converted directly to pyruvate. But ultimately, amino acids can be converted to lipid in a well-fed state. Sugar, glucose can be converted to lipid in a well-fed state. You store some glycogen. Urea is made because you get rid of the toxic alpha amino group from the amino acids that are coming from proteolysis. Some amino acids, of course, are also used in protein synthesis, and that protein is going to be such as the apolipoproteins that the liver does, plus a lot of other proteins the liver releases, some of them enzymes that help you, of course, digest food and then carry out the rest of the metabolic and immune processes. So the brain is going to be well served in terms of carbon source by glucose because it's water soluble, comes right into the blood, and there's a blood-brain barrier, so it's more difficult to get lipid into the brain. Okay, can come in the form of ketone bodies, which is later down the track for a discussion, if you have never heard of that before. The brain takes things like glucose all the way down to carbon dioxide and water. So it's a complete respiration, right? Completely oxidizes the reduced carbon and it completely reduces molecular oxygen, right? And that has to do with oxidative phosphorylation, electron transport chain, glycolysis, TCA cycle. I went backwards up that chain. All right. Urea is a degradation product and eventually it's going to be sent to the kidneys and excreted. All right, so you get the general idea. Lipid can be also sent to the adipose tissue. Major place for it. It's being located there or translocated there via these lipoproteins. The VLDL, very low density lipoprotein, and LDL play major roles there. Again, I'm going to go into a lot more detail of that later. Okay, but the lipoproteins are moving primarily in the lymphatic system. The other nutrients are moving in the direct bloodstream. Okay, but lipoproteins also are found in both places. Lipoproteins, of course, are also found in serum. All right. Red blood cells get fed lactic acid. The muscle tissue can store glycogen, and upon contraction in the muscle tissue, like skeletal muscle, you will also completely convert all the reduced carbon to CO2 and all the molecular oxygen, reduce that all the way down to water, right? But there's more than that involved. That the muscle also has a Cori cycle where it generates lactic acid, has an alanine cycle. It also can secrete glutamate and aspartate. Those are amino acids that get translocated back to the liver, used for gluconeogenesis, as can lactate. So there's a lot of things going on in the well-fed state, but I want you to get the idea that you have the basal metabolic functions going on, and you start to store a lot of depot fat. All right. Now, what about in the early fasting stage? The pancreas alpha cells now have to secrete a different peptide hormone called glucagon. By the way, the insulin was secreted from the beta cells, glucagon from the alpha cells of the pancreas. This is the component of the pancreas that has to do with endocrine metabolism. Pancreas also does exocrine metabolism, which we'll see later. The pancreas is really important here. Now, glucagon basically goes into the portal vein. What glucagon does is cause the breakdown of glycogen, so you get more glucose back in circulation, and is also involved in supporting gluconeogenesis, or the synthesis of glucose from non-carbohydrate precursors and non-lipid precursors in animals, right? All right, look what happens. Glycogen is broken down in the liver to glucose. That glucose then is sent to the brain directly. Of course, it's the early stages of fasting. The glucose also can make it to the muscle tissue, where it can get converted to pyruvate, and you're basically just carrying out glycolysis. The pyruvate then can go through lactic dehydrogenase, make lactate, sent back to the liver for gluconeogenesis, right? And then the glucose sent back out. You make very little glycogen in a fasting state, of course. Gut isn't involved because you're fasting, right? So it's a simpler system. Adipose tissue isn't being served by the lipoproteins, and they're not sending in much in the way of triglycerol or phospholipid or cholesterol to the periphery, because the body is concentrating on being homeostatic and regulating the amount of oxidizable carbon for the brain and the tissues, right? So this isn't entirely the case because, of course, there's always some trafficking of lipid, either from and in fact, the adipose tissue is going to be actively sending out, because of hormone-sensitive lipase, a lot of fatty acids going to go back into circulation onto the HDL, high-density-lipoprotein, and that fatty acid is going to be used in the liver to generate ATP because of beta oxidation of fatty acids making NADH and FADH2. Those two nucleotides then become oxidized in the electron transport chain, and you essentially then what? You make ATP, because the proton pumping ATPase in the inner mitochondrial membrane will use the reducing equivalents, the proton pumping, proton motive force in the inner mitochondrial membrane, the electron transport, of course, through the QH2 pathway, and ultimately through the four complexes of the inner mitochondrial membrane, and then ultimately protons moving back through the proton pumping ATPase, taking ADP and PI and making ATP. That's the ATP that's necessary for gluconeogenesis. So fatty acids don't give any carbon to glucose synthesis. They give the energy for it in the liver. Wanted to put that in there because it's not shown in this old Devlin slide. All right. Here are some metabolic interrelationships of major tissues in the fasting state. This gives you more information. It's basically what I just said. The muscle will break down protein into amino acids. Those amino acids, such as alanine, goes directly into gluconeogenesis to the liver. You can also, because of transamination reactions, right, make glutamine. Glutamine can make it all the way into the gut, and it can be converted to alanine. Alanine sent back to the liver for gluconeogenesis. That's through the enterocytic pathway. You also are sending some carbon always to the red blood cells because they're going to need it. And by basically all you're doing is taking reduced carbon, and usually red blood cells are just going to synthesize lactic acid, which can then again be gluconeogenic and sent back to the liver to synthesize glucose. Okay, protein is broken down in the liver. Glycogen is broken down. Ketone bodies. Here's the ketone body generation from adipose tissue. You're going to take fatty acids. You're going to synthesize acetoacetate and beta-hydroxybutyrate. Those are the ketone bodies that can serve as carbon sources in the established homeostatic fasting state, or moving towards the starvation state. So you start to shift from just glucose, because gluconeogenesis starts to decrease in the liver, and you start using more and more ketone bodies, because you're ultimately all the carbon you're going to use for the rest of the system when you increase the fasting into long-term starvation in humans, as long as there's adequate water and certain micronutrients are available, some of them which are called vitamins and minerals. Your body can last a really long time and live off the lipid reserve. This is how it functions. Now, you can get ketoacidosis with a high level of ketone bodies, but that usually doesn't happen in most people when they're dying of starvation. Usually what happens ultimately is just the carbon sources just get depleted, and you also become hypoxic, because a lot of beta oxidation of fatty acids means a lot of oxygen is getting used. So, and that's especially in the periphery, such as the skeletal muscle. Right? That's a different thing again, but you get the general idea. So everything that's here I've already talked about. You can also use glycerol. Remember, triacylglycerol, there is some carbon that can be used directly for gluconeogenesis. That's glycerol, and that's just going through the gluconeogenic pathway after making dihydroxyacetylphosphate, DAP, and also glycerol aldehyde 3-phosphate. And then it can re-enter gluconeogenesis and make glucose that way. Of course, the liver can take glucose 6-phosphate or glucose 1-phosphate, remove the phosphate, and allow glucose to be transported. Right? Most other systems do not transport glucose. So this is also what happens during the 5 phases of glucose homeostasis. The origin of blood glucose comes from exogenous at the beginning. That means your diet. By stage 2, this is hours after a meal, glycogen breakdown, and then hepatic gluconeogenesis starts increasing. It's where you start using amino acids from skeletal muscle, right, and the energy from the beta oxidation of fatty acids. More and more, as glycogen is gone, more and more gluconeogenesis. More and more is the glucose coming primarily from the liver and also from the kidney, that's hepatic and renal tissues. And then primarily, that's where all the gluconeogenesis is going to be coming from all the way to the end when you're way out here after like 40 days of fasting, right? Interesting, this shows you 40 days, right? Tissues using glucose, all of them use glucose, and then after a while, everything but the liver doesn't need anything because it's becoming a transporter of glucose. So muscles and the adipose tissue, increasingly, of course, the liver never does. More and more, what happens is the brain is the major utilizer of glucose, and that's what's showing here. The major fuel of the brain is glucose, and then when all the glucose is removed, more and more ketone bodies, and ultimately it's ketone bodies and whatever glucose is still in circulation because of degradation of glycogen from the periphery, and also still some gluconeogenesis coming from the liver and the kidney, right? So that's basically just showing you from a myopic glucose perspective. Right? All right. Now I want to talk a little bit about basic fundamentals in biochemistry. I've talked a lot about phosphorylation of enzymes and signal transduction cascades. One of the fundamental principles of this is that proteins can be phosphorylated. Okay, they can be phosphorylated from that anhydride phosphate from the gamma phosphoryl of ATP, right? Actually, the ADP can also transfer some of that same equivalent of a PI, or inorganic phosphate. Basically, though, what I want you to see here is that a non-phosphorylated form of an enzyme, via the activity of another enzyme or protein, any protein, via the activity of a protein kinase, kinases, those are the kind of proteins that phosphorylate, covalently modify other proteins. We'll take that gamma phosphoryl ATP and load it on, phosphorylate that enzyme. That enzyme then is phosphorylated. Many phosphorylated enzymes become activated when they're phosphorylated, and some of them just the opposite. They're deactivated, and some of them are just translocated, or they are aggregated with other proteins in the cell to form complexes which carry out the utility the cell needs to do. For example, autophagy or apoptosis, or maybe something like cell division, right? All right. So this phosphorylate, and then anyways, phosphorylated forms of proteins. There's a different enzyme called the phosphoprotein phosphatase. We talked about that, like P10 is one of them. And then you strip off the phosphate, and then the protein goes back to its native form. So if it was activated when it was phosphorylated, now it's deactivated, but still ready or poised to be reactivated. Sometimes it's degraded, depending on the activity and the biological potency of the protein. But the phosphatase is in control of that. So there are multiple regulatory pathways for the kinases. We talked about some of these, right? So such as signal transduction through an insulin receptor or a glucagon receptor or a catecholamine receptor, where the binding of free fatty acids is CD36 receptor, the orphan receptor. There's multiple ways that kinase cascades are turned on. The phosphatidylinositol 3 kinase pathway is one of them. Calcium storage being introduced also aids in the kinase pathways. Glucose uptake or glucose secretion from the liver, for example. All these things control phosphorylation cascades, which are all intracellular and therefore downstream what's going on in the plasma membrane in terms of the initial phase is sensu stricto of this signal transduction cascade. So now let's start looking at some hormones. This slide is showing you glucagon and epinephrine, okay? So also known as adrenaline. So glucagon and epinephrine stimulate glycogenolysis, the breakdown of the homopolymer of glucose, which is alpha 1,4 linked, 1,6 linked form of glucose, which is the branched glycogen. Starch is the same thing, only it has longer chains. There's two different kinds of starch, which you don't need to worry about right now. Amylose and amylopectin, for example. Anyways, you stimulate glycogen breakdown and you turn on gluconeogenesis because the cell is starting to starve. The brain wants to use glucose as the major energy source. Now, why is that? Why wouldn't fatty acid be the major energy source? Well, again, we've talked about this in various biochemistry lectures over the years, both online and in person in my classrooms. Basically, it's because the mobilization of lipids is slower and because the blood-brain barrier doesn't allow free access, so it's more difficult to get lipids sent to the brain. That's why ketone bodies serve as that carbon source when glucose stores are limited. Ketone bodies, of course, are oxidation products of fatty acids. Again, that's acetoacetate and beta-hydroxybutyrate, and they are quite water soluble. Right? So they're nothing like a long-chain alkane or mono or polyunsaturated fatty acid with a metal terminus and a carboxy terminus, respectively. So that's basically what I'm trying to tell you. Glucagon positively will stimulate an enzyme called adenylate cyclase. Epinephrine will also stimulate an adenylate cyclase, making a compound called cyclic AMP. There is a cyclic AMP dependent protein kinase, CAMP kinase, CAMP protein kinase. It's CAMP protein kinase A or PKA. And then it's going to carry out that phosphorylation. It's going to phosphorylate a series of enzymes, okay? I've given you the detail of which enzymes there are. There's a myriad of them, hundreds probably total. But right here we can say that some of the enzymes involved in glycogenolysis, like glycogen phosphorylase, some of the enzymes involved in gluconeogenesis, right? So Pep Carboxylase is one of those key enzymes, right? Those enzymes are going to be tuned up because they're phosphorylated, therefore activated. Yet enzymes involved in glycolysis, like PFK1 or glycerol 3-phosphate dehydrogenase, those enzymes, when they're phosphorylated, are shut down, shutting down glycolysis, because otherwise you'd have a futile cycle. In the same cell, you don't want to be synthesizing glucose, sometimes you're breaking it down. Although it's a rheostat model. There is a little bit of each, always. Likewise, though, if you're making glucose, you don't want to be lipogenic. You don't want to be making fatty acid. You don't want to be making prenolipids like cholesterol, because that's way too much energy necessary to do that, because it's reducing equivalents necessary to make carbon-carbon bonds in the form of what? NADPH from the oxidative pentose phosphate pathway, and also malic enzymes and other sources of the cytosol. So the enzymes involved in lipogenesis, like acetyl-CoA carboxylase, which is sort of the enzyme that synthesizes malonyl-CoA, which is the initial entry point for fatty acid synthesis for carbon, and you utilize that CO2, you release it, and biotin bound to it. And that was the first recognition what biotin does in the cell, because it binds carboxybiotin is how you're able to take CO2 and make a carbon-carbon bond with it, right? Biotin, that vitamin. At any rate, those enzymes which are lipogenic are turned down, glycolytic turned down, and the enzymes that are glycogenolytic and gluconeogenic are turned up by phosphorylation cascades. Okay. So you see that some are turned off, some are turned on, as I had hinted to. Now, the activation of the AMP kinase, which is a different enzyme, not the cyclic AMP protein kinase, but the AMP kinase, right? So the cyclic AMP protein kinase is called PKA, whereas this one's just called AMPK. So, for example, when muscle tissue, a lot of out there mowing the lawn, for example, or chopping wood, or cutting down trees, or in the wintertime shoveling snow, contractile activity of the skeletal muscle, doing a lot of work, you deplete the amount of ATP because you're stripping off that gamma phosphoryl, right? In so doing, you're generating energy because there's a lot of energy bound up in that phosphoanhydride bond. Hypoxia also will decrease the amount of ATP. Why? Because you need molecular oxygen in oxidative phosphorylation. You need to take O2 and reduce it all the way to H2O. Then the process, you take reduced carbon in the form of NADH, that's made after, say, glycolysis or beta oxidation of fatty acids, and then you're reoxidizing NADH and FADH2. Ultimately, those reoxidized nucleotides then generate that proton motive force to make ATP. Well, if it's hypoxic, you don't have any electron acceptor, because electrons are also flowing through that pathway. That's why it's called the electron transport chain in mitochondrial respiration and mitochondrial membrane respiration. Also, some of it happens in the peroxisome. In any way, if molecular oxygen touches decreased, it becomes hypoxic, you can't make ATP. It also happens with a lot of muscle activity. You become hypoxic. That's why muscles start to ache, okay? And you produce a lot of lactic acid. Lactic acid leaves the muscle, goes to liver gluconeogenesis. Cori cycle, I'm sure you've heard of this somewhere. Probably from me if you listen to my lectures, or from someone else. At any rate, what's going on here is AMP kinase is turned on when ATP is depleted. So what do you think AMP kinase is going to do, right? It's going to want to make more ATP, right? Yeah, because AMP, adenosine monophosphate, is after you've removed the gamma and the beta phosphoryls from ATP. So ATP to ADP to AMP monophosphate, you see? So when there's enough AMP, that kicks up AMP kinase. That turns on a series of phosphorylation cascades. That's going to cause glycolysis to tune up. The enzymes that are phosphorylated by the AMP kinase are going to be ones that are going to enhance glycolysis, not shut it down, because the serine or threonine or tyrosine residues on those enzymes involved in glycolysis are going to be embedded in different reaches of the sequence of those proteins, which are those enzymes involved in the glycolytic pathway, likewise for beta oxidation. And that also involves the carnitine palmitoyltransferase activity, acyl-CoA synthesis in the cytosol, acyl-carnitine synthesis in the mitochondrial membrane, and then all the beta oxidation enzymes, right? All of those are tuned up by phosphorylation by that enzyme, AMP kinase, at discrete sites in those enzymes. At the same time, you're oxidizing fatty acids, making a lot of reducing equivalents NADH, FADH2 to fuel, ultimately, gluconeogenesis. At the same time you're doing that, you're also making just a lot of ATP directly, right? Because you're oxidizing NADH and FADH2 via electron transport chain. So that's really important to understand. You don't always make ketone bodies, right? Sometimes you just take it all the way down to acetyl-CoA, and then that acetyl-CoA can make some ketone bodies, but it also could just be run through the TCA cycle, right? And also OAA is made, oxalic acid from pyruvate. All right, so you shut down gluconeogenesis. You don't need to make glucose in the starvation mode. Shut down protein synthesis that moves to the mTOR AKT pathway. It's just down lipogenesis. You don't need to make lipids when you need to make energy. And it's just down cholesterol genesis, which is another lipid, basically. Okay. Prenolipids, acelipids. Prenolipids are cholesterol type lipids because they work off isoprene, a C5 unit. And the lipogenesis that's considered with fatty acids is called acylip metabolism, because you make that radical form of that carbonyl, which allows you to be activated by a thioester, and that's how you make fatty acids. All right. Now, another detail about glucagon. Glucagon, as I told you, activates this adenylate cyclase, and there's a lot of detail about that, and I'll talk about that when it's necessary. But you increase the amount of cyclic AMP with that enzyme. That turns on PKA, as you know. Now that's going to be regulating at the transcriptional level the Krebs pathway. So you're going to have cyclic AMP response element binding protein, which is Krebs, okay? That's going to be phosphorylated. It's going to turn on glucogenic or gluconeogenic genes, enhancing gluconeogenesis, you see. Because that's the transcription factor that's going to turn on that DNA element that's a Cre element, right? So these are transcription factors or transacting factors, and that's a cis-acting factor because the Cre is basically a promoter region of that gene. At the same time, Krebs is going to negatively impact the transcription of all the lipogenic enzymes. So you have the same transcription factor. It's going to block the Cre genes, right, which are lipogenic. At the same time, it's going to activate the Cre genes, which are gluconeogenic. See how this works? Whenever one thing is turned on, the other pathway that could be opposing is turned down. Not completely off, just turned down. This is, again, a rheostat, right? Or a teeter-totter mechanism. Nothing is ever completely on or ever completely off. Cells can't do that because otherwise they would be at rest, and cells at rest are in morte. All right. What about fatty acids? I'm a lipid biochemist, so finally we get to talk about fatty acids. I mean, we have been already, but here's a little bit more front and center. Fatty acids work through the peroxisome proliferator activator receptor alpha and also a gamma form of that. And that's all positive. What that does as a transcription factor is it turns on the fatty acid oxidation genes. They're called FOX genes here. So that binding to its response element, the PPAR binds to its transcription factor, binds to the PPRE. We also have peroxisomal oxidation genes. Those are also important to turn on fatty acid oxidation. But in this instance, they're taking preformed phospholipids and phospholipids in the form of glycerol phospholipids and sphingolipids that are in the membranes, and they're breaking down some of the fatty acids that are in those phospholipids so that you synthesize new ones. The reason for that is because polyunsaturated fatty acids, which are in highly biologically active membranes, tend to become auto-oxidized. And they become auto-oxidized, and that could be detected by having a lot of free fatty acid, you see, because free fatty acid is actually toxic to the cell. Then you want to be able to use a peroxisome to get rid of that. Plus, the peroxisome is able also to shuffle everything through hydrogen peroxide and keep hydrogen peroxide from becoming toxic to the cell. I'll talk all about peroxisomal metabolism subsequently. All right. And there's also the ketogenic enzymes, right? The ones that make acetoacetate beta-hydroxybutyrate, right? Going through an HMG-CoA pathway, which is mitochondria. And basically then you turn on ketogenesis from fatty acids, and you're burning them up via beta oxidation. Beta oxidation of fatty acids is then synthesizing ketone bodies. Ketone bodies are then used as a source of carbon for the brain and all other tissues. Okay, so PPAR, that transcription factor, peroxisome proliferator activator receptor, works actually in the mitochondria and the peroxisome. It's first discovered in the peroxisome. That's why it's got that nice name. And there's an alpha form of it, isoform of it, is also a gamma form. And they do slightly different things in terms of transcriptional regulation. Now, here's something interesting. Okay, here's something now, and I'm going to diverge a little bit here. Talked about that. Here's a paper that was published just, 2 weeks ago. Actually, it's the 17th of April, 2020, so this wasn't quite 2 weeks ago. Published in a journal called Immunology Research. You can see the authors there. What is this paper talking about? Toll-like receptor 2. Now remember, TLRs are found on the surface of various cells, and they're usually involved in regulating some stress phenomena. Toll-like receptors are a component of the innate immune system. Okay? So TLR2, one of the isoforms of toll-like receptor, mediated metabolic reprogramming, participates in a drug called polyene phosphatidylcholine mediated inhibition of M1 macrophage polarization. Okay, now, why did this catch my attention? Well, it caught my attention because I wanted to show you an example about how papers just now published are still looking at things like PPAR. We're looking at PPAR alpha. So here we go. So we were talking about endocrine pathways. We're talking about single transduction cascades, phosphorylation cascades, transcription factors, beta oxidation, glycolysis, gluconeogenesis, all this cool stuff. Now I'm saying, okay, hold up. Dr. Guerra is just doing this plain old endocrine biochemistry lecture. Let's stop and let's look at some current literature. Okay, this is all, of course, got a teleological aspect to it. And you know that's the case because that's what I like to do with my lectures. So let's follow the Ariadne string, right? So PPAR alpha transcriptionally regulates the production of fatty acid beta oxidation genes. We just saw that in that old Devlin slide. Devlin writes a really good biochemistry book for medical students. Principles of biochemistry with clinical correlations you want to know. So M1, macrophage 1, pro-inflammatory macrophages. You got two types of macrophages. There's actually there's a suite of subclasses, but M1 pro-inflammatory, M2 kind of anti-inflammatory, at least contrary to inflammation, maybe not contradictory to it. So M1 pro-inflammatory macrophages work through this toll-like receptor 2, toll-like TLR2, to promote glycolysis over beta oxidation. Now look at that. And thus remain polarized as potent pro-inflammatory macrophages. Now these macrophages make it into a vascular bed. For example, the epithelia of lung. And these M1s are turned on, right? And they're turned on because what are they doing? They're not having fatty acid oxidation. Rather, they're using glucose as their major source of energy. They remain as an M1 polarity, which means they're pro-inflammatory, making pro-inflammatory cytokines, chemokines, transcription factors, growth factors, things like that I talked about in the past. So M1 pro-inflammatory macrophages work with TLR2. They promote glycolysis over beta oxidation, and thus they remain polarized as potent pro-inflammatory macrophages. Now, when PPAR alpha is activated, and we just saw it activated by what? By fatty acids, right? Yeah. But guess what? People have been trying to find pharmaceuticals that will activate it for various reasons. One is to diminish the pro-inflammatory response during disease state. What kind of disease state? Well, things like the constant hyperimmune state of type 2 diabetes, which is linked to obesity and aging, and both. When PPAR alpha is activated by a novel pharmacological agent, this polyene phosphatidylcholine. So phosphatidylcholine is a glycerophospholipid, has this polyene tail to it. It switches the polarization of those macrophages to M2 because you triggered PPAR alpha. And what's that going to do? You already know. Turn on the beta oxidation of fatty acids. So it's going to turn on ketogenesis, right? We just saw that. All right. But it just turns out that M2 polarization uses fatty acids as their carbon source rather than glucose. Why is that? It's because M2 macrophages have plenty of molecular oxygen, and they reside in an environment that has plenty of molecular oxygen and therefore not hypoxic, therefore not in a severely diseased state, which you find when M1 macrophages are triggering a hyper-inflammatory response. Now, why would that be? Why would you switch that glycolysis if you were a macrophage? Because a macrophage is going to a cellular environment expecting to do its own damage to destroy what? A pathogen, or an infected cell that has a pathogen, or a corrupted cell because it's mutated, or cellular masses in the lung, for example. I'm using lung only as an example. It could be the liver, kidney, heart, brain. Anyways, why would they want glycolysis? Because depleting oxygen is going to prevent that rapid amount of what? Cell division. It's going to kill things. Sometimes you need to kill what? Tumor cells. You need to kill virally infected, bacterially infected, or challenged, fungal infected, parasitically challenged cells. So when you do that, you're going to be in an environment already which has become oxygen depleted, because that's the kind of system which will shut down further cell division or growth of the pathogen. Viruses cannot transmit virion unless you have living cells. It's no good if the cells die because of lack of oxygen. Macrophages are cellularly aware of this. M1 polarization doesn't require molecular oxygen, so they make ATP from glucose. Hence, uptick in glycolysis. You see, there is a teleological perspective of all these biochemical pathways. And they more or less fit. Actually, they fit quite well. You just need to know everything to see how they fit. Now, when PPAR alpha is activated by that novel PC, as I said, it switches to M2, which means fatty acid oxidation, which also means lack of pro-inflammatory cytokines. It appears that fatty acid versus glucose oxidation, here's the take-home message from this paper, for bioenergetics pushes the M2 polarization, thus decreasing the pro-inflammatory status encountered in diabetic and obese patients. Okay? So if you already have a hyperimmune response, like an obese diabetic person, and you want to decrease the amount of that inflammation, let's say in in that same person that's getting a bacterial infection or maybe viral infection, this drug could be useful because it's going to be pushing to the M2 macrophages, which is going to suppress the local innate immune response. Inhibition of glycolysis with 2-deoxyglucose, which is a convenient way of using it, enhances the if I should use that for studying the movement or transport of glucose, because it's not metabolized. Inhibition of glycolysis with 2-DG enhances the anti-inflammatory effect of PPC. This drives, since this drives M2 polarity via PPR alpha, which is ligand activated transcription factor turning on lipid oxidation. We already know that, ligand being fatty acid, for example, right? And also through the TLR response that we were just talking about. Okay, now, so what is this paper showing you? That not only does this work well, it works much better. It enhances if you also kick down glycolysis. Kick down glycolysis, promote fatty acid oxidation, move to M2, decrease inflammation. You see. So inhibition of PPR alpha reduced the anti-inflammatory effect of PPC, of course, suggesting that PPC inhibits inflammation by reprogramming carbon source for bioenergetics, glucose versus lipids. A significantly downregulated expression of the sterol response enhanced binding protein 1C, okay, sterol receptor element binding protein 1C, right? So this is going to be working through a sterol pathway, it's a transcription factor. Significantly downregulated expression of that transcription factor occurs when you have a knockout mice on TLR2 and wild type bone-derived macrophages, okay? So that's what those are. BMDMs are bone-derived macrophages. So they're naive macrophages. They're neither polarized to M1 or M2. After exposure to PPC, which is the drug they're pushing here, that indicates a lack of dependence on TLR2 signals. So you don't need TLR2, okay? You still get a downregulated expression of that sterol pathway in wild-type bone marrow-derived macrophages. These are the naive macrophages when you give them the drug. So that means that TLR2 is just a convenient receptor here because it's often turned on with innate immunity. But it fulfills the teleological perspective that there are multiple aspects of metabolism to control even bioenergetics, which is considered by some to be dull and dreary biochemistry, thus regulating what? The difference between pro-inflammatory and anti-inflammatory responses in whatever tissue bed you're talking about. And so you don't need TLR2. It's just a convenient source when you have an innate immune response because it's binding to things like LPS and other like bacterial components, maybe viral spike proteins, although that's not the case with the coronavirus. There are some viruses that do trigger that. Okay. All kinds of other things trigger TLR2 too, as well, I mean. All right. So take a look now at insulin and glucose. So we just looked at that paper, and we're going to finish here with this. Insulin positively activates this SREBP, sterol receptor enhancer binding protein, 1C. It's a transcription factor. So it's going to be, that transcription factor is going to bind to the DNA in the nucleus of the cell that has an SRE, sterol response element, right? So sterol response element binding protein will be controlled by insulin via phosphorylation cascades because the insulin receptor is a autophosphorylating tyrosine system that phosphorylates other proteins. SREBP transcriptionally activates SRE genes, which are lipogenic genes, to increase lipogenesis. That's what insulin does. Insulin serves as the well-fed state, right? A lot of circulating glucose. Beta cells, the pancreas produce it. At the same time, it's going to shut down the fork head transcription factor. It's named after the Drosophila, which was first discovered. Mutations in the fork head transcription factor give you a fork head in Drosophila and in that fruit fly. So that fork head transcription factor then normally activates this IRE, which is an insulin response enhancer element. So normally that fork head transcription factor, when it activates IRE, would be giving you gluconeogenesis and fatty oxidation, but because insulin shut it down, see, that's what that means, that little club blunt. This doesn't happen, so you get a decrease in gluconeogenesis and you get a decrease in fatty acid oxidation, because that IRE normally controls gluconeogenic and fatty acid oxidation genes. Not happening now, okay, because insulin shut it down. You are getting lipogenesis because you're in a well-fed state, and glucose works through its own receptor system, okay, the CHR-EBP, and it co-enhances the steroid response receptor. And so it will generate more lipogenic genes at the transcriptional level. So you get two different ways. High circulating glucose, high circulating insulin work in tandem to increase lipogenesis, because all the carbon is going to be converted to triacylglycerol, ultimately leave a place like the liver and go to the adipose tissue and to the rest of the periphery via VLDL, IDL, LDL, right? The lipoproteins. Right? This is all good stuff in the well-fed state. Now, one more paper to discuss, and then we're going to stop. This is a paper published in Molecular Neurodegeneration, Volume 7, article number 45, published way back in 2012. When I say that, of course, I'm saying it, tongue-in-cheek, because papers that are published, even if they're published in the 50s and 60s, are still very good papers, as long as all the time since then, the research has shown them to be foundationally durable and accurate as it comes to the corpus of understanding we have in a particular scientific paradigm. So, from this paper published, which is not that long ago, 2012, the tumor necrosis factor and tumor necrosis factor receptor 1 dependent activation of sphingomyelinases triggers the production of ceramide, something we've been talking about in authentic biochemistry in both the video and the audio lectures. We talked about TNF being very important there. Produces ceramide, it's a lipid, sphingolipid, and other downstream lipid metabolites that promote the activation of the caspase, remember the cysteine aspartate proteases, 8/3, two different caspases, signaling, decreased AKT activation of mitochondrial membrane potential, and increased ER stress in dopaminergic cells, right? Dopamine responsive cells in the CNS. Remember, this paper was published in Molecular Neurodegeneration. Now, those dopaminergic cells, when they're degenerating, where do we hear from that from? What disease? Let me think. Parkinson's disease, that is correct. That's what this paper was actually looking at. So, from that paper again, ceramide causes apoptosis of the dopaminergic neuroblastoma, those like precursor cells, and also to neurons that are functioning in the dopaminergic pathway. Those are actually called the reward pathway in neuropsychiatry. Now, NSM or ASM, what does that stand for? Neutral sphingomyelinase or acid sphingomyelinase, something we talked about a great deal in authentic biochemistry recently. If you inhibit those during TNF treatment, it affords neuroprotection by blocking ER stress, we just talked about. Loss of mitochondrial membrane potential, caspase 3 activation doesn't happen, and decreases in AKT phosphorylation don't occur. You get neuroprotection because none of that happens because you're inhibiting, right, the sphingomyelinases, which work through tumor necrosis factor. I'm trying to show you a signal transduction cascade here. Why am I putting this all together now? So that you get an idea what's going on in the cells relative to bioenergetics and signaling. You get right down to it. Continuing. TNF treatment promotes a generation of ceramide and the accumulation of several atypical, that we don't normally talk about, deoxysphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphing sphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingos neuroblastoma cells, remember those are like naive, neuroblastoma cells are before you make the neuron, right? Dopaminergic neurons, to atypical DSPs, which are going to be made during TNF treatment, right? When they're exposed to these atypical DSPs in the micromolar range, which is pretty low range of biological activity, reduce cell viability, inhibited neurite outgrowth and branching. So you don't get good neurogenesis in these dopaminergic neurons. That suggests that TNF-induced de novo synthesis of atypical DSPs, which come from ceramide, may be a secondary mechanism involved in mediating neurotoxicity of a tumor necrosis factor and its receptor-dependent activation of the sphingomyelinases, because that's what's being triggered to make ceramide and these DSPs downstream, right, that promote degeneration and, of course, the caspase-dependent programmed cell death of the DA neurons. All right. So we're going to stop there. This paper here was just published about a year ago in the Journal of Biological Chemistry. And it's going to talk now. We're going to switch from, we're still all in the endocrine mode here, right? All I'm going to do is I'm taking these papers from the primary literature to give you a complete understanding of how the neuroendocrine and the plastic endocrine system, which comes from all the organs involved, like the pancreas and the liver and the kidney and the brain, all those endocrine organs are in the adipose tissue, how all of that's going to fundamentally maintain a homeostasis, which can be perturbed because of its poise upon stress. And so the stress we've been pointing at here this morning has been that associated with obesity. Remember the free fatty acid story, obesity being a major disease in the human population, and which is also a very potent contributor to lots of other diseases, which don't normally associate with obesity. What kinds of diseases? Well, we know the ones that do: cardiovascular disease, atherosclerosis, but also cancer. Also, the way the body reacts to pathogen invasion. What kind of pathogens? Bacteria, pathogenic parasites. Most parasites are pathogenic. Fungi, as well as, viruses. All right. So we are going to stop here. And we're going to continue. 15 minutes was a good signal for this lecture today. Again, this is Friday, the 17th of April, 2020. And the next lecture, we're going to go further into detail of this endocrine pathway. I've got how many slides? 132 slides. I won't do all of those, but I'll do a lot of them because I'm going to do multiple lectures on this to give you what people have been asking for, this foundational understanding of biochemical systems in humans, so that you can then interpret better the current scientific literature. And rather than leaving it at the end, 6, 7 lectures later, I bring in these papers all the time right now, because you people aren't afraid of good research articles. Heck no, because you're authentic biochemistry subscribers, or you want to be authentic biochemistry subscribers, or subscribers to VeraMed, or to my YouTube feed, or just go to my Facebook. So, anyways, this is Dr. Dan Guerra coming to you from the Authentic Biochemistry VeraMed studios in the beautiful inland Pacific Northwest, now in the state of Washington, where we reside. And what I will say to you is have a pleasant Friday afternoon, and bye for now.